organic compounds required as nutrient in tiny amounts by an organism. An organism, of course. Yeah, it's an error. Vitamins cannot be synthesized in sufficient quantities by an organism and must be obtained from the diet. Here, body can synthesize some amount of some vitamins, but it is not enough. So there is a must to get vitamins from food. Vitamins can be classified as lipid soluble and non-lipid soluble or hydrophilic soluble or hydrophilic vitamins. Lipid soluble vitamins are found in the fats and oils of food. They are absorbed into the lymph and carry the blood with protein transporters. They are absorbed together with chylomicrons. We remember about lipid lipoprotein metabolism, where we have seen that lipids from the gut are absorbed as chylomicrons. So chylomicrons contain also lipid-soluble vitamins. As hydrophobic compounds, they are absorbed efficiently with lipids. This means that fat or oil must be consumed for the vitamins to be absorbed by the body. Vitamins are stored in liver and body fat and can become toxic if large amounts are consumed. Lipid-soluble vitamins are stored in the liver and body fat, and in the case of excess, they have toxic effects. You see, they are different from minerals because minerals are inorganic, but vitamins are organic. You see, vitamins, we have lipid soluble and hydrophilic. Those lipid soluble are stored in liver and body fat. And as they are organic compounds, they can be changed, activated, reduced, or fail, or whatever. While you have seen that minerals remain unchanged. Now, we also have water-soluble vitamins or hydrophilic vitamins. Those vitamins dissolve easily in water. They are found in vegetables, fruits, and grains, and meat. They are absorbed directly into the bloodstream. They are not stored in the body, and the toxicity is rare. Alcohol can increase the elimination of such vitamins. Also, smoking cause decreased absorption. When you compare lipid-soluble and water-soluble vitamins, you see lipid-soluble vitamins can be stored in the body and they are associated with toxic effect, while water-soluble vitamins are not stored, so there's no, the toxicity is rare, and water-soluble vitamins are affected by alcohol and smoking. Now, among water-soluble vitamins, we have vitamin A. No, I'm sorry. Now, vitamin A, there are 3 different forms of vitamin A. We have retinol. This is an alcohol. It has alcohol function. You have retinal, aldehyde, and retinoic acid. And these are known as retinoids. You see, when you say retinoids, you are mentioning 3 forms of vitamin A, and all those 3 forms are active. The cells of the body can convert retinol and retinal to the other active form of vitamin A as needed. Each form of vitamin A performs specific tasks. You see, retinol supports reproduction and is the major transport form of vitamin. Retinal is active in vision and is also an intermediate in the conversion of retinol to retinoic acid. Retinoic acid acts like a hormone, regulating cell differentiation, growth, and embryonic development. Once again, vitamin A is present in 3 forms with different function. We have a retinol. This is an alcohol function. Here, retinol is shown. We have retinal. It is an aldehyde. It is shown here. And we have retinoic acid. This is trans and cis retinoic acid. And you have seen that they have different function, or they function differently. And where retinol supports reproduction, retinal in vision, and retinoic acid is a hormone regulating cell differentiation, growth, and embryonic development. As I was saying, we are talking about water-soluble vitamins. The first is vitamin A. Here we have structure. Now, vitamin A precursors. Let's see the precursors of vitamin A. Foods derived from animals provide retinol in a form that is easily digested and absorbed. The main dietary sources of vitamin A are the precursor vitamin A carotenoids from fruits and vegetables. The body can convert carotenoids like beta-carotene, alpha-carotene, and beta-cryptoxanthin into vitamin A. When carotene is split, at least one intact molecule of retinol or retinoic acid can be obtained. You see, carotenoids from fruits and vegetables we consume. Once in the gut, it can be dehydrolyzed or cleaved, either to give retinol or retinal or retinoic acid. Just here to show source of vitamin A and beta-carotene. Vitamin A comes from animal sources such as eggs, meat, and dairy products. Beta-carotene, a precursor of vitamin A, comes from green, leafy vegetables and intensely colored fruit and fruits and vegetables. You know about carrots. All these are pictures indicating the source of vitamin A. Let's see vitamin A transport and metabolism. From the gut, carotenoid absorption is by passive diffusion. It is converted to retinol, then in the retinol esters and join chylomicron in enterocytes. Almost all retinol absorbed into the enterocyte leaves via the lymphatics as retinal esters in chylomicrons, then move into the general circulation where they are hydrolyzed and reach the liver as chylomicron remnants. Almost all retinal esters present in the chylomicrons remain with the particle during conversion of chylomicron remnant. Here we have a picture.
Extra question.
Question.
I think chylomicron isn't it a lipid?
Oh no, you remember chylomicron in
Cholesterol.
in cholesterol metabolism.
Yes.
You remember in the part of chylomicron, hydrophilic component are on the surface, while hydrophobic are inside. Now, this vitamin A absorbs as carotenoids. In the gut, they are hydrolyzed to form retinol. And this retinol, it is an alcohol. You remember esterification reactions. There is a reaction where retinol join with acid to form esters. Once they form esters, they join chylomicron. So they absorb together with chylomicron as they are esters. And those esters will be inserted inside the chylomicron, while on the surface you have cholesterol and phospholipids, whatever, all esters are inside. So chylomicron, those vitamin A are absorbed with this chylomicron. And here we have intestinal lumen. This is in the intestine. This is beta carotene. Upon hydrolysis, we have retinol, and retinol, as it is soluble, is the water soluble. You know, they are absorbed. They enter by diffusion into the enterocyte. In the enterocyte, also beta carotene can be enter the enterocyte. There is this retinol formed form esters. Retinol esters join chylomicron to enter blood circulation. And what is written here is what is shown here. And just the following is the mechanism associated with this the full metabolism of vitamin A. Let's see here extrahepatic uptake of chylomicron remnant. You remember, chylomicron as it passes through the blood, what is hydrolyzed is a triglyceride until the remnant is taken by the liver. Now, from here, this is intestinal mucosa, showing that retinol esters are formed and enter the bloodstream as chylomicron. Phospholipids, the lipase act on chylomicron, removing fat or removing free fatty acid. Then the remnant is taken by the liver. What is taken in the liver is retinol esters. When retinol esters enter the liver, they are hydrolyzed back to form retinol. And in the liver, there's different kind of cells. There is also cells called satellite cells. Now, those stellate cells, inside the stellate cells, retinol esters are stored. Now, with the association with this retinol binding protein, also retinol can reach the blood circulation and enter other or extracellular extrahepatic cells. Extrahepatic cells mean cells or other organs than the liver, where the vitamin A is needed. Now, on this picture, what is written here? It is also chylomicron remnant are mainly cleared by the liver. Extrahepatic uptake of remnant may be important in the delivery of retinol and carotenoid to extrahepatic tissues, such as bone marrow, peripheral blood cells, spleen, adipose tissue, skeletal muscle, and kidney. Now, hepatic processes of chylomicron remnant in hepatocyte include the secretion of retinol binding protein. We are here. All this is in the liver. The secretion of retinol binding protein. Sorry. No. Bonjour, where's Sari Kondahuzi? Yeah. So I'm teaching, I'm the class. Sorry. I was saying that hepatic processing of chylomicron in hepatocyte include the secretion of retinol binding protein, which bind retinol and deliver it into the circulation, then to extrahepatic cells. About 90 to 95% of the liver retinol is stored in a liver cell called stellate in the form of retinol esters packed together in the cytoplasmic lipid droplets. Now, here we are trying to show major pathways for retinoid transport in the body. From here, once again, we start with absorption in the intestine, enterocyte, lymph duct, circulating in this is chylomicron remnant in the blood circulation, reaching the liver. Here we are in the liver, where they are converted into retinol binding protein. It is transferred to peripheral tissues. Also inside in the liver, we have stellate cells where this retinol is stored in lipid droplets. Now, this retinol binding protein together with retinol is sent to the peripheral tissues, binding to different kind of with the involvement of different kind of protein, if I can say. This TTR means transthyretin, RAR retinoic acid receptor, this RX retinoid X receptor is a kind of receptors on the peripheral cells where the complex will bind, then internalized for further processes. Just here, these are, it's a kind of summary to show from the gut to the liver to peripheral tissues, and the indication of involvement of different factors. Here there is a protein, transthyretin, also receptor. This transthyretin function as a receptor, also as a binding protein. Once they bind, there is a receptor, if I can say, or a doubt, I don't know. It binds on the cell surface, then enter for further processing. You see, in the further process, there is also involvement of gene expression. When this retinol bind on the complex enter, there is retinoic acid in cis retinoic acid, which will act on the gene or the DNA to express needed protein or needed activity. Just here there is a text indicating what was shown here. When you try to read, it is written that dietary retinol esters are hydrolyzed retinol in the intestinal lumen prior to absorption by enterocytes, and the carotenoids are absorbed and then partially converted to retinol in the enterocyte. In enterocyte, retinol is esterified to fatty acid before incorporation into chylomicrons. Chylomicrons then reach the general circulation by way of intestinal lymph, and chylomicron remnant are formed in blood capillaries. Chylomicron remnant, which contain almost all the absorbed retinol, are mainly cleared by the liver, parenchymal cells, and to some extent also by cells in the other organs. In liver parenchymal cells, retinol esters are rapidly hydrolyzed to retinol, which then bind to retinol binding protein. This complex is secreted and transported to hepatic stellate cells. Stellate cells may then secrete retinol RBP directly into plasma, and most of this complex in the plasma is reversibly complexed with transthyretin. As I was saying, in the circulation, it is complexed with this transthyretin and remain in the blood, of course. The uncomplex retinol is presumably taken by a variety of cells by cell surface receptors specific for this protein. Most of the retinol taken up will then recycle plasma either on the old binding protein or bound to newly synthesized protein. Here, just to show from here, absorption, retinol esters, chylomicron, lymph, circulation, then hydrolyzed in the capillaries to have this remnant. Remnant can come directly to those cells. Also, this can mainly enter in the liver. It is converted back into retinol. Retinol bind to binding protein. This binding protein can transfer it into stellate cells for storage, or can leave and enter the bloodstream. Now, in the bloodstream, now this complex can also taken back by parenchymal cells of the liver, or can bind to transthyretin, and this complex circulating the blood, it can come back or hydrolyzed back to form transthyretin and retinol, or continue in this direction where this complex binds to receptor, then formation of this 9-cis-retinic acid, then induce function in the cell, depending on the cell and the need of the cell. Metabolic function of vitamin A, regulation of gene expression. We have seen that this 9-cis-retinoic acid, how it acts as activator or transcription factor, regulation of gene expression. Vitamin A is a part of visual pigment rhodopsin, maintaining clarity of cornea. Yes, eating carrot is good for your eyesight, of course, because of carotenoid present in carrots. Vitamin A is required for cell growth, division, cell growth and division for epithelial cells, bone and teeth. Vitamin A promotes development of immune cells, especially natural killer cells. Plays a role in mobilizing iron from liver stores. So vitamin A deficiency may also compromise iron status. It has antioxidant activity. Excessive intakes of preformed vitamin A can result in high levels of vitamin in the liver, a condition known as hypervitaminosis A. Now we finish with vitamin A. That's the pathways. Let's see deficiency. Since vitamin A is stored in the liver, it may take up to 2 years for signs of deficiency to appear. Night blindness and very dry, rough skin may indicate a lack of vitamin A. Other signs of possible vitamin A deficiency include decreased resistance to infections, tooth development, and slower bone growth. These are conditions associated with vitamin A deficiency. Toxicity of vitamin A. Symptoms of vitamin A toxicity include dry, itchy skin, hair loss, headache, nausea, and loss of appetite. Signs of severe overuse over a short period of time include the dizziness, blurred vision, and slowed growth. Vitamin A toxicity also can cause severe birth defects and may increase the risk of hip fractures. It also causes liver enlargement. We said before that those lipid-soluble vitamins can be stored in the liver. So the high amount of those soluble vitamins, there will be a high amount stored in the liver. So this will cause liver enlargement, hepatomegaly, because of high content of vitamin stored in the liver. Another lipid-soluble vitamin is D. Are we together?
Yeah.
Yes.
Okay. About vitamin D, with the help of sunlight, vitamin D is synthesized by the body from a precursor derived from cholesterol. Vitamin D is not an essential micronutrient because given the right season and enough time in the sun, we can have vitamin D. Now, the active form of vitamin D is actually a hormone that targets organs, most notably the intestine, kidney, and the bones. We have seen it. In the intestine, vitamin D is involved in the absorption of calcium and phosphorus. In the bone, It assists in the absorption of calcium and phosphorus, helping bones grow denser and stronger as they absorb and deposit these minerals. Now, the active form of vitamin D, once again, is calciferol. Now, vitamin D is a steroid. It contains steroid nucleus. This is cyclopentanol hydrophenanthrene ring. This one, sorry. Vitamin D in diet occurs in two forms. There is ergocalciferol and cholecalciferol. We have D2 ergocalciferol and D3 cholecalciferol. Ergocalciferol is formed from ergosterol and is present in plants. Cholecalciferol is formed from cholesterol and is present in animals. Cholecalciferol and ergocalciferol are sources for vitamin D activity and are referred as provitamins. Just to show the structure of vitamin D2 and vitamin D3. Formation of vitamin D during the course of cholesterol biosynthesis. 7-dehydrocholesterol is formed as an intermediate. On exposure to sunlight, it is converted to cholecalciferol or vitamin 3 in the skin. Just here to show, this is the skin, the sunlight, and this 7-dehydrocholesterol is converted into vitamin D3. Also, vitamin D3 can be taken from food like fish and meat. This D2 can be taken as supplement, also enter the liver. Now, in the liver, this cholecalciferol is, we have seen that it is a provitamin, is converted into the hydroxy vitamin D3. And in the kidney, there is also addition of hydroxide group, dehydroxy vitamin 3, which will maintain calcium balance, and this is calcitriol. Absorption that from animal source such as animal liver contain vitamin D3, that from plant sources contain vitamin D2. Absorption vitamin D and D3 are absorbed from upper small intestine and the bile, and the bile is essential, so we need bile to absorb vitamin D. Vitamin D3 and 2 from mixed micelles by combining with bile salt, and those micelles are presented to mucosal cells to be absorbed by passive transport. We still consume D2 and D3. Now in the gut, they are mixed with bile acid, then absorbed as the micelles enter the cells by passive transport. There is a vitamin, a binding protein, remember, binding. But before that binding, here, let's see this vitamin D binding globin, which will bind a protein, a globin protein, to be transported. So vitamin D is transported from the enterocyte binding on this globin protein. This vitamin D hydroxylated on 25 carbon, and the one hydroxylated on carbon 1 and 25, they are transported in the blood, binding to vitamin D binding globin. And it is this 25 hydroxycholecalciferol is the major storage and circulatory form of vitamin D. What I'm saying here is that for the vitamin D to be transported in the blood, it needs a binding protein. And the binding form is in two forms, this 25 hydroxy and 125 dihydroxy. All those two forms are bound on the binding globin, but to be stored, it should be 25 hydroxy cholecalciferol, and of course they are stored in the liver. Metabolism and biochemical functions. The synthesis of the hydroxycalciferol. Let's see, this is the active form. The active form of the vitamin D is 125 dihydroxycalciferol and is also called calcitriol. Cholecalciferol is first hydroxylated at 25 position. It becomes hydroxycholecalciferol. There is an enzyme called hydroxylase. This enzyme is present in the liver. Now from the liver, this 25 hydroxycholecalciferol reach the kidney, where there is an enzyme specific to hydroxylate 25 hydrocholecalciferol at carbon position 1. This is another hydroxylase in the kidney. There is an hydroxylase in the liver and another hydroxylase in the kidney. So what is absorbed is this. It is still not final. So this cholecalciferol, once it is in the liver, it's hydroxylated, then circulating the blood to reach the kidney, then another hydroxylation. What is important here is to show the role of this hydroxylase, because any impairment in the hydroxylase will affect availability of vitamin D. This enzyme found in the liver require cytochrome P450, NADPH, and the molecular oxygen for hydroxylation process. So the hydroxylation process in both liver and kidney needs cytochrome P450, NADPH, and the molecular oxygen. Now, the formation of vitamin D is regulated by regulation of renal hydroxylase. And the activity of this enzyme is increased by hypocalcemia. Do you remember? Low level of calcium affect parathyroid hormone. Parathyroid hormone activate vitamin D or accelerate vitamin D production. You see, this parathyroid hormone will act on the liver and kidney, and it is the target is this alpha hydroxylase, which need to be activated to make a final form of vitamin D or a functional form of vitamin D. So this enzyme is indirectly affected by hypocalcemia. Now, hypocalcemia stimulates this parathyroid hormone secretion, which in turn increase hydroxylase, I said it, and this hydroxylase may be feedback inhibited by the presence of cholesterol. High level of cholesterol will inhibit this 1-alpha hydroxylase in the kidney. Clinical importance: in chronic renal failure, where this enzyme activity is decreased, it leads to the decreased synthesis of this vitamin. This condition will lead to renal osteodystrophy or renal rickets. It's called renal rickets because the problem is the kidney. You may have rickets due to the problem of the kidney, where the kidney cannot make this enzyme responsible for activation of vitamin D. And you have seen that without the vitamin D, there will be this will they will have impact on the bone density. This is a condition. This condition is treated by giving vitamin D preparations. If there's an individual having this problem, an individual will need to get a supplement. This enzyme, the enzyme deficiency, can also occur as inherited disorder or due to
Teacher, question.
regulation.
Does that mean that the ricket can be treated?
If it is renal rickets, if the problem is the kidney,
Yeah.
there is a way to get this supplement.
But I think also this, if it is the renal ricket, we must have some deformation on the.
Is associated with renal failure. Renal failure is irreversible. There is a stage where you can reverse, but if it becomes irreversible, there is no way. But if there is a way to reverse the condition, it be very early, there is a chance to reverse the condition. But if this is not inherited, if the problem is acquired, the condition is treatable to prevent further complications. But you cannot say that already deformed individual will see a child with rickets and you say, I'm going to give him or her this vitamin D, and you believe you will reverse the condition. It will not work. This is treatable when you know it is manifested very early, and you know exactly that the problem is there associated with the kidney. Now, the condition can be treated in this way. Okay?
Yes.
Okay. In the regulation of plasma calcium and phosphorus, this is a repetition of what you have seen in the mineral metabolism, where vitamin D regulates the plasma levels of calcium and phosphorus. Plasma calcium levels are regulated by effect of this vitamin D on the small intestine and the kidney and the bones. It maintains the plasma calcium levels by increasing absorption of calcium from small intestine, increasing the absorption of calcium by renal distal tubules, and increasing mobilization of calcium from bones. Ibi twabibonye. The same here, that it function at 3 different levels to maintain plasma calcium. Action on intestine, let's repeat it. When repeated many times, you get more understanding about it. Now, cholesterol increases the intestinal absorption of calcium and phosphate. In the intestinal cells, cholesterol bind with the cytosolic receptor to form a cholesterol receptor complex. This complex interactive with specific DNA leading to synthesis of specific calcium binding protein. This protein increases calcium uptake by intestine. The mechanism of action of calcium is similar to that of steroid hormones. You remember steroid hormone, how they induce gene expression. Now action on bones in osteoblasts, bone formation. Calcium stimulates calcium uptake for deposition as calcium phosphate. Calcium is essential for bone formation. Calcium, along with parathyroid hormone, increase the mobilization of calcium and phosphate from bones, causes elevation in plasma calcium and phosphate. Now on kidney, it is involved in minimizing the excretion of calcium and phosphate through the kidney by decreasing the excretion and enhancing the absorption. You have seen it. Reason why I will not spend much of time on it. We have a metabolite of vitamin D. Remember, vitamin D is 1,25-dihydroxycholecalciferol. But here we have an intermediate metabolite where we have 24 and 25-dihydroxycholecalciferol. This is synthesized in the kidney. There is another kind of hydroxylase hydroxylating the structure on carbon 24. Calcium concentration is adequate. When calcium concentration is adequate, this hydroxylate acts leading to the synthesis of less important compound to maintain calcium homeostasis. Now you see, this is the mechanism of regression. You see, this calcium concentration is adequate, means there's no need of further production of this hormone. In the case there is no need formation of this calcium, but you still have 25-dihydroxycholecalciferol what? So for regression, there is formation of less important compound where instead of phosphorylating carbon 1, carbon 24 is phosphorylated. And this is a mechanism to maintain calcium homeostasis. Are you together?
Yes.
Yes, we are.
Okay. Try to talk to me to be sure that we are together. Don't get tired. I want you, especially for who is a student, to finish this topic by today. Deficiency of vitamin D cause rickets in children and osteomalacia in adults. You have seen it. This rickettsia is the vitamin D deficiency state in children. It is caused by dietary deficiency and non-exposure to sunlight. It is characterized by bone deformities due to incomplete mineralization. Deficiency of vitamin D will cause enlargement and softening of bones. It will cause delay in teeth formation. The weight-bearing bones are bent to form bow legs. There will be decreased serum calcium, deformation of muscle due to weakness of abdominal muscles. Just to show the case of rickets, you see, this is the problem of deficiency in vitamin D.
Lecture question. My question is concerning about the exposure to the sunlight. Approximately what time and the duration really good or appropriate to exposed to the sunlight.
What is the question?
The question is to know the real time and the duration of time.
Okay. Theoretically, it is said the morning sun is very important in vitamin D formation for children. So the sunlight which is not intense, the morning one. I don't know the theory behind, but at least 1 hour on the sun. I don't know exact time is for overexposure, but what is I know is the morning sun, and the exposure should not be too long, approximately 1 hour, like that. We may read about. I'm not sure.
lecture ni nko guhera saa kumi n'ebyiri kugera nka saa mbiri n'igice.
Yes, the morning sun, saa mbiri saa tatu gutya.
Kariya akazuba wicaraho ukumva utakavaho.
Kariya kakaba gakenewe cyane 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 ku bana. Si byiza gufata umwana ngo uhore umukingiranye mu nzu atajya ku kazuba, burya baba bari gukora amakosa.
Ku bantu bakuru ariko lecture bikoze cyane wahinduka imbwa.
Of course umuntu ukuze ko waramaze gukomera ubu ntabwo nawe urikeneye, ariko ntabwo urikeneye nk'umwana. Twabonye ko calcium is needed, highly needed for a growing child, kuko nk'aho amagufa yamaze kwiforminga already. Osteomalacia, vitamin D deficiency in adult, it is caused by inadequate exposure to sunlight or low dietary intake. It is with the osteomalacia, there is a demineralization occurring in spine, pelvis, and lower extremities. Bowing of long bones may occur due to weight of the body. Flattening of pelvis bone may cause difficulty during labor. All these are problems associated with vitamin D deficiency in adult. About renal rickets, in chronic renal failure, 1-alpha hydroxylase activity is decreased, leading to decreased synthesis of vitamin D. The condition leads to renal osteodystrophy. A condition is stated, this is a repetition. No need. Vitamin D toxicity. Vitamin D is stored mainly in liver. It is most toxic in overdoses. Toxic effects include the mineralization of bones and increased calcium absorption from intestine, leading to increased plasma calcium, hypercalcemia. You know, hypercalcemia is associated with the deposition of calcium in many soft tissues, such as kidney and arteries. It leads to formation of stones, calcicular. High consumption is associated with loss of appetite, nausea, increased thirst, and loss of weight, etc. Now, vitamin E. We are talking about lipid soluble vitamins. Vitamin E is chemically known as tocopherol. Tocos, childbirth, phyllo, to bear, and O, alcohol. This is an alcohol because you see the hydroxide group attached here. An alcohol was capable to prevent reproductive failure in animals. This is alcohol is known to prevent reproductive failure in animals. So it is thought as anti-sterility vitamin. Chemistry of this hormone, vitamin E is the name given to a group of tocopherol and tocotrienols of this structure. We have about 8 tocopherols, but the most active is the alpha tocopherol. So vitamin vitamin E is fortified cereals, seed and seed oils like sunflower, nuts and nut oils like almond and green leafy vegetables. These are sources of vitamin E. Vitamin E is absorbed along with fats in the upper small intestine. It is combined with bile salt to form mixed micelle and taken up by the mucosal cells. In the mucosal cell, it is incorporated into chylomicron, the same as vitamin A. Vitamin E is incorporated to chylomicron and enter the circulation. In the circulation, chylomicron transport vitamin E to the peripheral tissue or to the liver. In the liver, vitamin E is incorporated to very low density lipoprotein. You know, very low density lipoprotein cholesterol is the one synthesized in the liver and released into the circulation. In the circulation, VLDL is converted to low density lipoprotein cholesterol. Finally, vitamin E is transported with low density lipoprotein cholesterol to reach the peripheral tissues, including adipose tissue. This vitamin is stored in liver and adipose tissue. It is present in biological membranes because of its affinity to phospholipids. The biochemical function of vitamin E. Most of the function of vitamin E is that is related to antioxidant activity. It has antioxidant property. It prevents non-enzymatic oxidations of various cell components by molecular oxygen and free radicals such as superoxide and hydrogen peroxide. Selenium helps in these functions. Vitamin E is lipophilic in character and is found in association with lipoproteins, fat deposits, and cellular membrane. Just to say that vitamin E is associated with lipids and preventing lipid peroxidations because it acts as scavenger for free radicals. This is the main function of vitamin E. It is an antioxidant and it functions in association with selenium. This is we read about selenium in a trace element. Vitamin E protects polyunsaturated fatty acid. You know, those acid with many double bonds. You know, double bonds are very sensitive to free radicals. So the presence of vitamin E will prevent membrane peroxidation or lipid peroxidation, especially the target Z biological membranes. So this vitamin E is essential for membrane structure and integrity of the cell. It is a membrane antioxidant. It prevents the peroxidation of polyunsaturated fatty acid. It protects red blood cells from hemolysis by oxidizing agent. It is associated with reproductive function and prevents sterility. I don't know how. This is another topic. How the mechanisms of this vitamin to prevent sterility. I don't know many about it. It preserves and maintains germinal epithelium of gonads for proper reproductive function. Maybe this is the point where vitamin E intervene in the prevention of sterility because it protects germinal cells. Here, mini gametes or gametocytes. It increases the synthesis of heme by enhancing the activity of enzyme delta aminobutyric acid synthase, and you remember those enzymes in porphyrin metabolism. Now, the vitamin is required for cellular respiration through electron transport chain. It stabilizes coenzyme Q. You remember in biochemistry when we were talking about about electron transport chain and oxidative phosphorylation. Now, vitamin E prevents the oxidation of vitamin A and carotene in its function of antioxidant. It is required for proper storage of creatine in skeletal muscle. It is required for absorption of amino acid from intestine. It is involved in synthesis of nucleic acid. It protects the liver from toxic compounds such as carbon tetrachlorides. It works in association with vitamin A, C, and beta carotene to delay the onset of cataract. You see how important is vitamin E. Vitamin E is recommended for the prevention of chronic disease such as cancer and heart disease. Vitamin E prevents oxidation of low-density lipoprotein cholesterol. The oxidized LDL promotes heart disease, you know. So this vitamin E will prevent this oxidation. When we say LDL is atherosclerotic lipids, it is because once it is oxidized, will favor the position of lipids in arteries, promoting atherosclerosis and the heart disease, associated heart diseases. So this vitamin E is very important in preventing this oxidation. It is important in preventing heart disease associated with lipid metabolism. It is a component of this selenium is a component of glutathione peroxidase and functions as antioxidant. Now, selenium is included here because it has the same function as vitamin E. Vitamin E and selenium work in the same way. The main purpose or the main function of them is to prevent oxidation of important biomolecules. Vitamin E reduces the requirement, this selenium reduces the requirement of vitamin E in diet. It is required for normal pancreatic function and enhancing the absorption of vitamin E. Now we finish with vitamin E. Now we are on vitamin K. Vitamin K is a group of hydrophilic or hydrophobic vitamins. We have K1 and K2. K1 comes from plants. K2 is produced